Welcome to the first lecture that is part of Biology 100. This is Chapter 1 from your Concepts of Biology. The material in here will correspond to what is in your textbook. This first chapter is looking at an overview of biology. It starts here with this picture of Earth from a NASA image from a satellite, just to kind of show you that biology really does encompass the entire planet. And there are lots of different things that have become possible for us to do because of studying biology that involve this planet. Biology is the study of life. We don't have any magical ingredient that makes something alive. So instead, what we do is we look at the properties of life to determine whether or not something is alive. When you're looking at things that are alive, sometimes these things look pretty straightforward, but other times it can seem to be a little bit of gray area with these. When we see things that are alive, they're always going to have order to them. It is a predictable order that you see within a species. If you look at animals, we see a similar pattern with the animals all having two eyes. They're on their face. They have a nose, a mouth, all of those things on the face. They have limbs for mobility, upper limbs and lower limbs, or front limbs and hind limbs. The order is consistent. Living things are going to respond to stimuli, so they will react to their environment, changes that go on around them. Living things are also able to reproduce. You don't get life without life. One of the main things of the cell theory is that all cells come from living cells. All life is made of cells. Living things are also going to adapt to their environment. This adaptation can be rather quick in some cases. Other times they can adapt slowly, more of evolution over time. Living things are going to grow and develop. Most living things do not begin life as a full-size individual. They will start out smaller and grow and develop to become the adult full-size version. They will regulate their internal systems. Most living things are also going to try and achieve homeostasis, which is maintaining a consistent environment. One easy way to look at homeostasis is we like to keep homeostasis with our body temperature. If we start to get too warm, we're going to sweat to try and accommodate that to cool ourselves off. If we are cold, we're going to start to shiver to try and generate some more heat. And then all living things are going to need to have a source of energy and process that energy to make it a useful form to carry out life's activities. So for example, you have a toad here. This is an example of being highly organized. He's made up of cells. The cells are going to come together to form tissues. The tissues come together to form organs and the different organ systems within the toad. So for example, his digestive system has order to it. It's going to be made up of multiple different organs, all looking to get nutrients into his body. This is an example of a plant here that is sensitive and going to respond to his environment. The leaves will actually fold up when you touch this plant. Here we have some examples of things being carried out, shared characteristics. With this group of kittens, they've all inherited genes from both parents, so they're going to have a lot of similarities to them, but they're also going to have unique features as individuals where they are each their own unique combination of the genes from their two parents. The polar bear here is an example of a mammal that is able to do homeostasis to maintain their body temperature. They're going to generate heat through muscle use. They're also going to reduce heat loss through having thick fur, lots of body fat underneath their skin. And here an example with a condor. This is an animal that's going to require the use of a lot of energy in order to be able to fly. They're going to derive that energy from chemicals that they get from food. And it's just an example of how different life forms use each of these processes. So this here is a model of DNA, your deoxyribonucleic acid. This is composed of the different atoms. This is going to be your genetic information. This is going to carry the instructions for making every structure in every cell of all living organisms. So when we look at the levels of organization of life, the chemical level is the most basic. We start out with atoms, which are the smallest unit of matter. You can break atoms down into subatomic particles, but they will no longer retain the identity of the particular element they are if we break them down more. So we consider them to be the smallest unit of matter. A molecule is made up of two or more atoms. Sometimes in chemistry they like to specify with a molecule that it's two or more atoms of the same element. In biology, we don't really focus too heavily on that definition. A lot of times people will talk about water molecules, and water is made up of two different elements, oxygen and hydrogen. And our molecules will come together to form our macromolecules. These are the large molecules that are going to be combined of several smaller units called monomers. And we will look at those in more detail in the next chapter. On the cellular level, our cells are going to fall into two main categories: the prokaryotic cells or prokaryotes, and eukaryotic cells or eukaryotes. Your prokaryotic cells are going to be the simpler cells. They do not have any specialized organelles or membrane-bound compartments. What they do have is genetic material and a plasma membrane around the outside. Your eukaryotes are going to be more complex. They're going to have membrane-bound organelles. The organelles are specialized structures within the cell. When we look at the level of organization going on to a much bigger global portion, we start out with the chemical level as the most basic. The chemicals will come together to form cells, or the cellular level. Obviously there is something more. You can't just take chemicals together and make something living. Again, there's that magic ingredient that we don't have an explanation for why something on one hand is a cluster of chemicals that is alive and why another cluster of chemicals is not. The cells will come together to form tissues. Those are just groups of cells working together. The tissues can come together to form organs, which are more specialized. They're going to have different types of tissues working together. Your organs come together for organ systems. The example I used before, the digestive system, all of those different organs in the digestive system are working together for the common goal of providing nutrients to that organism. The organism is the total living individual. A population is going to be a group of individuals of the same species that are living together, where a community is going to be a group of different species all living together. The ecosystem is going to take into account all of the different communities as well as the non-living factors. And the biosphere is the entire portion of the earth that supports life. So this here just gives you a visual demonstration of those different things, going from, at the top, your atomic level, the molecules, the organelles, the cells, tissues, organs, and organ systems, the organism, the ecosystem, and the biosphere, which includes the entire planet. When we look, there is a lot of diversity in life. And there are several reasons for that. That's part of what we will be looking at, is all the diverse types of life that come about. Evolution has allowed us to have this diversity. It's a process of gradual change during which new species arise. Evolution is a constantly going on process. We've constantly been gaining new species, and we're always losing some species. The idea is that hopefully you are not losing them faster than you are gaining them, which is what we anticipate is actually going on the planet currently. Taxonomy is the study of naming organisms. What's been used for several years is the Linnaean system or the Linnaeus system. This is going to organize things by the domain, the kingdom, phylum, class, order, family, genus, and species. The domain is going to be the most encompassing, and the species is going to be the most specific. When we talk about a particular organism, we will use the binomial nomenclature using genus and species together. When we look at the domains, there are 3 domains of life. There's the archaea, the eukarya, and the bacteria. Both the archaea and the bacteria are going to be prokaryotic cells. The eukarya are going to be eukaryotic cells. So most of these you've probably heard of before. The archaea may be a group that is new to you. And we determined these 3 domains of life by looking at the sequences of the ribosomal RNA in their cells. The archaea, although they are prokaryotic cells, do share about as much similarity with the eukaryotes as they do the bacteria. Another way of looking at them is phylogeny. Phylogeny is going to be a way of organizing the different organisms to help show the evolutionary relationships in a diagram format. So this is showing the normal taxonomic hierarchy for a dog, from the broadest category, the domain, down to the most specific, down at the bottom, the species. So you'll notice down there at the bottom when you look at it, under species, you see Canis lupus. Above, for genus, you see Canis, because genus and species are used together for the binomial nomenclature system. So this image here just represents some examples of the different domains of life. Image A is showing some prokaryotic cells, some bacteria. Image B is showing some of the archaebacteria. They're what we call extremophiles because they live in extreme environments. And here they're showing them in some hot springs. C, you've got a sunflower here, eukaryotic cell in the plant world, and then you have the lion that's part of the domain eukarya. Both the plants and the animals are eukarya. This is an example of a phylogenetic tree. This was constructed by Carl Weiss using genetic relationships. Some of the phylogenetic trees that we had in the past are being updated as we start to be able to sequence the genomes of more organisms. So you'll see there's 3 main branches: the bacteria, the archaea, and the eukarya. So when we look at the branches of biology, there are several. These are just a few examples. Microbiology, that focuses on things that are microscopic, looked at underneath the microscope. So a lot of bacteria studied in that field, as well as other microscopic single-celled organisms. It also includes organisms that are a little bit larger but have a microscopic phase, such as the parasitic worms. Molecular biology looks more at the molecular processes. Paleontology, zoology, botany, ecology, biotechnology, genetics are all some examples of different fields of biology. So this is showing here some researchers working on excavating dinosaur fossils. So it is still a field of biology. This is showing a forensic scientist working on DNA extraction. So forensics uses a lot with DNA these days. So here we're looking at some, in picture A, some cyanobacteria underneath the light microscope. And then in picture B, you've got some stromatolites along the shores of Lake Thetis in Western Australia that are created by cyanobacteria. And these are examples of microorganisms at work. This is an E. coli picture underneath an electron microscope. A lot of bacteria do look very similar underneath the electron microscope, so we have to use other means to actually identify them. This is using a scanning electron microscope, so you can see the outside surface and gives more of a three-dimensional type of image. So biology is just one field of science. There are other fields in science: astronomy, computer science, geology, logic, physics, chemistry, mathematics. Some of them will use more math, where others will do more observational type of studies. Science is just knowledge of the natural world, and the natural sciences are going to be the fields related to the physical world and its processes. So things will tend to get divided into physical science and life science. Biology is definitely in the field of life science. Your physical science would include more of the things like chemistry and physics. To study science, we've come up with the scientific method, so we have a means of finding reliable, consistent, accurate information that everybody understands how to do. And the scientific method works really well for that. It was not put together lightly. There are three main areas that we look at in there with the scientific method. Your hypothesis, you've probably heard it being described as an educated guess. And that's really kind of oversimplifying the hypothesis. Although it is a guess, it's more of a tentative explanation rather than a guess. It's more researched than just randomly guessing for something. Scientific research is expensive to do, so to be able to conduct experiments, you're generally not going to get funding to do that unless you've got a lot of good background information that you have very good reason to suspect your hypothesis or your explanation is going to be true. So there is a lot more that goes into forming the hypothesis than just coming up with a guess. Scientific theory is going to be something that's well tested. So in everyday language, when we use the term theory, you may be talking with your friends and you'll say, hey, I have a theory as to why this happened. In everyday language, the term theory implies a lot of doubt. In science, a theory is well tested, not a lot of doubt. You don't get to be a theory without having a lot of testing from multiple people coming in. Scientific laws, these are extremely well tested, and these have been observed over and over and over. So in order to change a scientific law, it would be something very major would happen. An example would be the law of gravity, and it's going to apply universally in our environment here on Earth. Now we all know that when you go out into space, there is not gravity there. It isn't that it doesn't apply, there is explanations for it, but the law of gravity is going to be a factor into everything we study here on Earth. So Sir Francis Bacon is actually the one who is credited with being the first to document the scientific method. So when we do scientific inquiry, we have inductive reasoning and deductive reasoning. When you use inductive reasoning, you're going to use correlated observations to arrive at general conclusions. Deductive reasoning is going to use general principles or laws to explain specific results. You have to be careful in using inductive reasoning. If you look at too many specifics and try and come up with the big picture, you may make false assumptions. That would be like if I were to look around in a classroom and I would see that in my classroom that day, all the males are wearing hats. So I would use inductive reasoning to say, all right, if there's an individual wearing a hat, it's a male. Those observations may hold for my particular classroom on that particular day, but that inductive reasoning can actually be really faulty and get me in trouble because, as we all know, not all males wear hats at all times. So we generally try and use deductive reasoning when we can. When we create a hypothesis, we have to test it. One of the things that you have to have your hypothesis be is falsifiable. You have to have a way of disproving it through experimental results. When you design your experiment, you're going to have variables. These are any part of the experiment that may change. Ideally, we want to try and limit the variables as much as possible. If you have too many variables that can change, it becomes very difficult to determine is something causative in nature. The control are going to be parts of the experiment that cannot change. So this is just kind of a graphic overview of the steps that we go through in the scientific method. First, you're going to be making observations, and then you will ask a question. Most of the time, this is going to lead to making more observations, and finally forming a hypothesis that will answer that question. At that point is when you're going to make predictions based on the hypothesis, do a test, and then analyze the results. Most of the time, you are not going to have things go the way you want the first time. Sometimes the hypothesis will be supported and you report the results. A lot of times the hypothesis is not supported, so you have to go back and try again. It's not unusual for people to spend the majority of their career trying again and again and again before they actually are able to answer some of the questions they may come up with. When a hypothesis is actually able to go through this process, you get the results you were predicting. It's actually pretty significant in the field of science. So just coming back to those variables, with the variables, there are going to be some of them that you are going to change, and there are going to be some of the variables that you are going to look to see if they have changed as a result of what you've done. So when we look at science, basic science or pure science aims to expand knowledge. It's basically studying science for the purpose of expanding your knowledge. Applied science is what technology is. It aims to use science to create solutions to real-world problems. One of the ways that scientists will document their work is through peer-reviewed articles. The idea of them being peer-reviewed is that if other experts in the field look at their work, they're able to go through, critique it, look for possible errors and sources of mistakes, and help to improve the work overall. One example of a major project that was taken on was the Human Genome Project. This was completed in 2003 and started in 1990. So this was a collaborative effort among many researchers working in several different fields of science. Originally, it was just those working on sequencing the genome manually using techniques in the lab. By having several different fields of science come together in there, including those with computer applications, they were finally able to automate the process. It was anticipated that the Human Genome Project, when it was first started, would be something we would still be working on well into the year 2020, and it was completed much quicker as a result of the collaboration. So there are some additional videos and links that will help provide additional information to get you going on the overview.